Alright, so I'm going to be talking about mitosis firstly. So the first thing I want you to notice is that this is not mitosis, this is interphase, and this is where the cell spends 90% of its time. The nucleus is made up of chromatin, which is 50% DNA and 50% protein. And this is the nuclear envelope, the nucleolus. The nuclear pores are the holes in between the nuclear envelope, and this is an animal cell, obviously. These two things are called the centrosome, which are made up of a pair of rods held perpendicular to each other. These are gonna be quite important in mitosis later on. So the first stage of mitosis is prophase 1. This is where the nuclear envelope and the nucleolus disappear. So as you can see, there's a thinning out of the nuclear envelope and there's no nucleolus anymore. The centrioles are separated and they move to opposite poles. This, you can see it moving right down to the opposite side. The spindle fibers form and they attach to the centromeres of the chromosomes. So yeah, the centromere is what's holding the two sister chromatids together, and the chromosomes have shortened and thickened. So this is the second stage of mitosis, which is called metaphase. Metaphase, the chromosomes will line up randomly in single file on the equatorial plate. As you can see, there's a line that causes them. This is done by the spindle fibers, which are these thin things coming out, pushing and pulling the chromosomes. Right. So the third stage of mitosis is anaphase. This is when the spindle fibers pull on each identical sister chromatid and they separate them to the opposite poles. And this is telophase. So the nuclear envelope begins to reform, and then the spindle fibers disappear. The chromosomes lengthen and thin out, and the cleavage furrow forms, which is only in animal cells. As you can see, they are two identical daughter cells. And then this is the final stage of mitosis, which is cytokinesis. So as you can see, these are two distinct cells. Mitosis has occurred, and it's 2N to 2N, and they are somatic cells, and it's an equal separation of everything, including organelles and all that. So that is mitosis. Meiosis is quite a little bit more complicated. So yeah, let's start with that. So prophase 1 of meiosis. So interphase is essentially the same thing for meiosis, so there doesn't need to be an image shown. Okay, so for prophase 1, the centrioles and asters move to the poles, just like in mitosis. The spindle fibers form, just like in mitosis. And then the homologous chromosomes line up to form a tetrad. So 1, 2, 3, 4, that's a tetrad because 4, obviously. And this is called the homologous pair. Those two together, that's a homologous pair, and that's a homologous pair. So where they line up to form a tetrad is called the synapses, and this synapses allows this is where the chromatids overlap and it's called the chiasmata, sorry, and it allows for crossing over of genetic information. So as you can see here, it's now the blue has crossed over, and these ones are still like just normal, but these have crossed over, which allows for greater diversity of offspring. And yeah, it's the first stage of meiosis 1. So meiosis metaphase 1 is the second stage. So the homologous pairs line up on the equator, the equatorial plate, and random assortment. So they line up randomly, which is also for genetic uh, genetic diversity, I guess. So yeah, they pretty much just do the same thing. They line up with their sister chromatids. And so this is the third stage of meiosis, which is anaphase 1. So the homologous chromosomes separate, and each chromosome move towards the opposite pole. It's also known as segregation. As you can see, they have both separated. And in telophase one, the nuclear envelope and the nucleolus form, and then cytokinesis happens, so we get two distinct cells. So this is the first stage of meiosis 2, which is called prophase 2. Yeah, essentially it's the same things that happen as in prophase 1. The the chromosomes shorten and thicken, the nuclear envelope begin to disappear, the asters move to opposite poles, and yeah. So in metaphase 2, they line up randomly at the equatorial plate, and they are getting pulled by the spindle fibers. They're getting pushed and pulled to line up properly. And then for anaphase 2, the seventh stage of meiosis, they separate the sister chromatids. So now there's across the equatorial plate, there's this is going to be a distinct cell, this is going to be a distinct cell, and same as those other two. And then for telophase 2, what we have is the cleavage furrow, obviously. And then what we have here is these are going to be two distinct cells. The nuclear envelope is beginning to reform. The nucleolus has reformed. There's one aster on each one. It's going to develop later on when it's ready to do meiosis again. And this cell went from 2N to N. And this is meiosis pretty much. Yeah. And then at the end, we're going to have four distinct cells with, yeah, that's meiosis.